In order to control and integrate the numerous body functions, body organs, body systems that we have, the nervous system has to have a wide variety of synapses. Now, each type of synapse can be different in terms of its neurotransmitter or its receptor. And each type of receptor can have a different outcome or a different result in the target cell. Now, I've drawn 3 different examples of synapses on the board behind me just to illustrate some of the differences that you might see, but there are many hundreds, if not thousands, of types of synapses in our nervous system. So, the 3 that I'm going to share with you are the excitatory cholinergic synapse, the inhibitory GABAergic synapse, and the excitatory adrenergic synapse. First, I want to break down these terms. Excitatory means that the postsynaptic cell, the postsynaptic neuron, will depolarize or become closer to having an action potential. Inhibitory means that the target cell is less likely to have a response, less likely to have an action potential. The second word in each of these synapses refers to the neurotransmitter that's being used. So for instance, this first one is cholinergic, and it gets its name because the neurotransmitter is acetylcholine, or ACH, as I've written down here. So cholinergic, acetylcholine. GABAergic, the neurotransmitter, is GABA, which stands for something that I don't even remember, gamma aminobutyric acid or something like that. The third one is adrenergic, and the adrene here should remind you of the word adrenaline, which its proper name is epinephrine. And this kind of synapse uses norepinephrine, a form of epinephrine, which is abbreviated NE. Okay, so let's take a look at what happens in each of these different synapses. Here, the presynaptic neuron is filled with, or not the whole neuron, but the axon terminus here is filled with acetylcholine containing vesicles. When the action potential arrives at the end of the neuron, at the end of the axon, these vesicles release acetylcholine. Acetylcholine binds to its receptor. This is exactly like the examples we've used so far. Binds to its receptor, which happens to be on a ligand-gated sodium channel. Sodium, when the ligand binds, it opens the channel, so sodium enters the cell, making the cell more positive or depolarizing it, and that's how we get a local potential. If it reaches the threshold, then this cell will have an action potential. Now, we call these, and this is important for the next lecture, EPSPs, or excitatory postsynaptic potentials. Excitatory because we're getting closer to an action potential, postsynaptic because we're in the postsynaptic cell, and potential because we're talking about differences in charge across the membrane. In the next example, when an action potential arrives at the axon terminus, the vesicles that are releasing neurotransmitter release the neurotransmitter GABA. GABA binds to its receptors on ligand-gated channels. However, in this case, the channels are chloride channels. Now, chloride happens to be similar to sodium in that it's most concentrated outside of the cell, which means that when these channels open, chloride rushes into the cell, making the inside of the cell more negative. And you can see here that the cell became more negative. We call this hyperpolarization because it's even more different from the outside. It's more negative compared to the outside. And whenever a cell is hyperpolarized, it's more difficult for that cell to reach an action potential or to have an action potential because it would require more sodium from another synapse in order to bring the membrane potential up to the threshold. So we've moved away from the threshold. We've made it harder for this neuron to have an action potential. So we've inhibited it. And it's therefore called an inhibitory postsynaptic potential. Now, the third kind of synapse I have here, the excitatory adrenergic, an action potential arrives at the axon terminus. The vesicles release the neurotransmitter norepinephrine. Norepinephrine binds to its receptor on a different kind of sodium channel, and that depolarizes the cell. But in this case, instead of the cell having an action potential, it causes something called cyclic AMP. This is a special kind of nucleotide, similar to or related to adenosine triphosphate, except this is adenosine monophosphate, and it's chemically arranged so that it's cyclical. That's where it gets its name. This cyclic AMP acts as a second messenger and goes into the cell and causes activation of different things or inhibition of different things. So it can activate enzymes, or it can activate transcription and translation of genes. So these are just 3 examples of synapses, and any one postsynaptic neuron could have multiple types of synapses connecting with it. And that's what we're going to talk about next, is how does that postsynaptic neuron decide whether to have an action potential or not, especially when it has many synapses.